Okay, in this video, we're going to talk about fermentation, the process in which glucose is broken down into pyruvate, and then that pyruvate is going to be further metabolized to make acids and alcohols. This is what is happening in your kombucha and is also what happens in your cells if there's not enough oxygen to do regular aerobic respiration. So the first thing that I want actually you guys to remember is when we do aerobic respiration, the first three steps, glycolysis, pyruvate oxidation, and the Krebs cycle, are all involved in breaking down the glucose. So in each of those steps, we're breaking carbon-carbon bonds, releasing electrons, and releasing the excess single atom carbon as carbon dioxide. So when carbon-carbon bonds are broken in this process of aerobic respiration, carbon dioxide is produced and also electrons are released. I am not going to put numbers with these because I don't actually want you guys to focus on balancing the equation. What I want you to focus on is when bonds are broken, electrons are released. Okay, that's what we're going for. The reason that this is important is these electrons are going to be carrying with them the energy that we need to do stuff, like make ATP. And so we cannot let these electrons just wander around the cell willy-nilly. Instead, we are going to capture those electrons in an electron carrier. So in the three processes that we learned about, glycolysis, pyruvate oxidation, and the Krebs cycle, the most common electron carrier is NAD+. FAD can also be used, but the most common is NAD+, so we're going to use it in our little example. So the electrons are going to go to the NAD+ to be carried, and then this will make NADH. Okay, everybody good so far? So when we break bonds, we release electrons that can be used to do stuff. The successful bond breaking and capturing of the released electrons depends on there being NAD+. Everybody with me so far? So what happens in glycolysis, right, is we have our 6-carbon glucose. We're going to break this bond here to make 2 3-carbon pyruvate. We're going to make some ATP, and we're going to make some NADH, because here we've had to add the NAD+ in order for the NAD+ to capture the electrons that are going to be released when this bond is broken. Okay, in order for glycolysis to happen, we have to have NAD+, because NAD+ picks up these electrons when this bond is broken. If this bond does not get broken, nothing happens at all. No glycolysis, no pyruvate oxidation, zippo. So this is a pretty important reaction, and this reaction happening depends on there being NAD+ in the cell. Okay, everybody good so far? The reason that this is important is once the NAD+ has accepted electrons, it turns into NADH. Okay. So we need some way for the NADH to give back its electrons to somewhere in order to reform the NAD+, because it's like a cycle, so that the NAD+ can be reused here in glycolysis. If we are doing aerobic respiration, which we talked about last time, then we take the NADH, we donate the electrons to the electron transport chain, thereby remaking the NAD+. Eventually, these electrons are going to go to oxygen in order to make water. In the process of the electron transport chain, they will provide energy to pump the protons across the membrane. That is what will be used to make ATP. But the bit that I want you to get here is that in aerobic respiration, the NAD+ is regenerated in the context of the electron transport chain, which requires oxygen. So the problem that we have is how to get back the NAD+ if we do not have any oxygen. Okay, so cells will do fermentation either when there is straight up 0 oxygen or not enough oxygen to replenish the NAD+ at the rate that is needed. Either of those, so 0 or limited oxygen is going to be when fermentation happens. Okay, let me just get a new sheet of paper here. Ta-da! Okay. So in fermentation, and again, this is when we're going to have no or limited oxygen. So you might have learned fermentation always happens when you have no oxygen. I mean, kinda. So you have no oxygen available right then to accept the electrons, but maybe there was oxygen like 5 seconds ago. There's just not right now. So fermentation can also happen in a limited oxygen situation, just when there's not enough oxygen for the electron transport chain to function. So there's two basic steps in fermentation. Step one is glycolysis, which is the same exact glycolysis that you learned about before. So we go 1,6-carbon glucose to 2,3-carbon pyruvate. In the process of that, we make 2 net ATP and we make NADH. I'm going to be straight with you. I can't actually remember the number of NADHs that are made. You'll have to watch the respiration video for that. We make some number of NADHs. Okay, so the second step is going to be NAD plus regeneration. I do not know actually what Dr. Knight in your book call this. They might call the second step fermentation. That's kind of confusing to me because the process is called fermentation, and so I go with the second step being NAD plus regeneration. Okay, so at the end of glycolysis, we have these three things. So we've got the NADH, and we are desperately wanting to give its electrons to somewhere. So we've got the NADH, and we would really like to shove the electrons off in order to remake the NAD+. But where can we shove the electrons to? That is our question. ATP does not take electrons. So we then have the pyruvate, a carbon thing, which we know takes electrons because it just gave up some electrons. So what happens in NAD plus regeneration is we have the NADH and we have the pyruvate. The electrons from the NADH go to the pyruvate. So that makes then NAD plus, and this is the a little bit tricky part. The carbon product varies depending on what the enzyme is that is used, which varies depending on what the type of cell is. And so in your kombucha jar, when the yeast do NAD plus regeneration, they make alcohols like ethanol, etc. When bacteria do NAD plus regeneration, they make acids. And so that's why the pH of your kombucha jar is so low. And so the process of fermentation can produce alcohols and or acids depending on the identity of the cell that is doing it and depending on what enzymes are present there. Okay, so we have the pyruvate is 3 carbons, right? If we add some electrons, then we're going to do some rearranging. The alcohol might end up to be 2 carbons, acetic acid is 2 carbons, so we've actually lost a carbon, but the important thing is that we are not totally down to CO2. And if you guys will remember, we talked in the respiration video about how there's electrons that make up the bonds, and the energy associated with those electrons is what is used to do stuff. And so these carbon compounds have not been completely broken down to CO2. There will actually also be CO2 produced, which is where the third carbon goes and is why your kombucha is fizzy. But there is still 2 carbons stuck together, and thus there is still some associated energy. And so what happens in your kombucha is you have glucose goes to pyruvate, goes to alcohol, and this is the yeast. And then that alcohol can be used by the bacteria to make acid. Alternatively, we can go directly from pyruvate to acid by the bacteria. But what I want you guys to understand here is that alcohols and acids, actually depending on the microbe, can be energy sources because they still have 2 carbons. But actually, I think that might be a little bit sidetracked. So to recap, the reason that we have to do NAD plus regeneration is to remake the NADH, or the NAD plus. Then that NAD plus is going to come back up here to glycolysis and allow the cell to run glycolysis again, definitely generating these two net ATPs, but possibly also allowing the cell to proceed to pyruvate oxidation, the Krebs cycle, and eventually electron transport, which is going to be where the bulk of the ATPs are made. But if glycolysis not running without the NAD plus, and the NAD plus, in the absence of sufficient oxygen, the NAD plus cannot be made without NAD plus regeneration. And so the point of fermentation is bizarrely not to make alcohol. The alcohol is just a waste product, actually. The point of fermentation is to remake the NAD plus in order to allow glycolysis to proceed. Fermentation happens entirely in the cytoplasm of the cell and does not require oxygen because there is no electron transport chain present. So glycolysis, cytoplasm, fermentation, NAD plus regeneration also happens in the cytoplasm and does not require oxygen. And the point of NAD plus regeneration is to remake the NAD plus so that glycolysis can be run again by the cell.